Thank you, Gabby, for your nice introduction, and thanks to the organizer of the Dazzle series for giving me the opportunity to present my my preliminary data on a new project that I started in the lab of Dr. Puri at the Sanford Barnard Brevis. So before moving to the the science, I just want to describe, so what is Duchenne muscular dystrophy? Duchenne muscular dystrophy is a very debilitating disease that affects mostly the peripheral muscle of the human body. And you can see that it destroys the structure, the nicely tied, organized structure of the human muscle with this infiltration of fibrotic tissue, adipose tissue, and all these little cells that are inflammatory cells. It's a rare disease and with a symptom of onset in early childhood. And unfortunately, the kids that are affected by Duchenne muscular dystrophy are wheelchair bound by their early teens, and their life expectancy is around 20 to 30 years. There is a milder phenotype of the disease that is called Becker muscular dystrophy. Both diseases are caused by a mutation in this huge protein that is called dystrophin, which is located on the sarcolemma of the muscle fiber and organize the dystroglycan complex that was shown to have a main function to make the muscle resistant to contraction. So the continuous contraction of the muscle fiber will not destroy the cells. But it has also additional function that are important to mediate the mechanical stimuli of the cells feels from the outside to the nucleus by two different pathways. On one end, you have the mechanotransduction that transmit these forces, contraction forces, from the outside to the nucleus. And on the other hand, dystroglycan complex binds and inactivate via the nitric oxide synthase HDAC2, preventing the translocation to the nucleus. When dystrophin is absent because it's mutated, all dystroglycan complex is absent, and so both of these mechanisms are inactive, and you have an aberrant gene expression. So, but what is important to know, especially for my talk and in general, is that the genome is not linear. Even if we remember the classical textbook, biology textbook of a genome, we have the gene with some promoter, announcer, different gene, and we see it as a linear. So, but this is not the functional version of the genome, because according to whatever cell type or whatever stimuli the cells receive, this linear genome folds and acquire a different conformation, in which in this case, the announcer interacts with the second promoter to activate this gene, and it's not able to interact with this promoter to activate another gene. While in a different cell or in the same cell, but with different stimuli, the announcer is now able to interact with the first promoter to activate the first gene, but not anymore with the second promoter and gene. So in these two cases, we have a completely different transcriptional output. So since we know that dystrophin can mediate the signal transduction from outside the cells to the cell nucleus, we had two questions. The first one was, can the lack of dystrophin in Duchenne muscular dystrophy change the genomic conformation? And if we restore dystrophin in Duchenne patient, that is the cure that is proposed and it's now in clinical trial for the disease, are we able to reacquire the healthy conformation, or we still have some pathogenic conformation and therefore some pathogenic gene expression? So to address this question, we developed two different in vitro disease models. in which we took either the fibroblasts or the iPS that were donated from Duchenne muscular dystrophy patient, and we were able to transdifferentiate them into myoblasts that are the progenitor cells of the skeletal muscles, and the myotubes. And as you can see from the staining, we have, in all cases, we are able to obtain these elongated multinucleated myofibers that are positive for the expression of myosin heavy chain. And you can see there is no different efficiency in the system between wild type or Duchenne. So when we investigated the expression of a gene as TGF-beta1, that is a profibrotic gene that is upregulated in dystrophic patient, in these myotubes, in these in vitro generated myotubes, you can see that independent of the underlying mutation, we have an increased expression of TGF-beta1 compared to the control myotubes obtained by healthy cells. And when we check the three-dimensional conformation of the TGF-beta1 locus, we saw that in healthy myotubes, the promoter interact with this regulatory element to block the expression of TGF-beta1. But in the DMD myotubes, we observe a lack of interaction between the promoter and the regulatory element, and TGF-beta1 is still expressed. So if we restore dystrophin, are we able to restore the gene, at least the gene expression of TGF-beta1? So in collaboration with Chamberlain and Olson labs, we decided to restore dystrophin using two different approaches. One of them is in clinical trial, that is the upregulation of functional dystrophin protein in the myoblast by using of viral vector. In this case, we use lentivirus, and we infected either the myoblasts or the myotubes to have corrected myotubes. And in the other case, we decided to use genome editing using the CRISPR-Cas9 to correct the mutation of dystrophin in the cells from the patient to obtain a functional version of the protein. And also in this case, then we obtain corrected myotubes. However, when we check the expression of profibrotic genes such as TGF-beta1 and a pro-inflammatory genes such as CTGF, that are both upregulated in the disease model, you can see here and here, in none of the corrected version of the myotubes are we able to go back to similar levels seen in wild type situations, suggesting that even if we are able to restore dystrophin in the myotubes, we still have underlying pathogenic conformation of the genome that sustain this pathogenic expression of this gene. So in the future, we want to confirm this finding that using genome-wide unbiased analysis, such as RNA-seq or ATAC-seq or iChip, to see on the three-dimensional conformation of genome. And additionally, we want to test other drug therapies such as that are in clinical trials, such as the HDAC inhibition, in conjunction with dystrophin restoration to see if we are a better output. And obviously, we want to move from the in vitro dish model to an in vivo studies using the mouse model of Duchenne muscular dystrophy. So, but what about diversity? I moved to San Diego from Italy 4 years ago with my beautiful husband. And if some of you are from UCSD and had kids at the preschool of UCSD, you might know him very well since he worked there for a couple of years. And I am quite vocal sustainer of the LGBTQ issue in trying to fight the LGBTQ facing issue in academia. So, but before going to the issue that LGBTQ people face in academia settings, let's go through a few definitions, because words are important and words have meaning. And so I would skip the most common terms, but let's just focus on a few few terms like transgender, that is a person that whose gender identity differs from what is typically associated with what their sex that was assigned at birth. And sometimes you are this word cisgender. So cisgender is just anyone that is not a transgender. And then you have two other words, gender nonconforming and gender queer, that if you just look on the definition, you might feel that are synonymous with transgender, but they're not. And you should, if someone define themselves as gender nonconforming, you should not call them transgender or genderqueer. And basically, to summarize, you can think that queer as an umbrella term that covers all the other terms, and lesbian, gay, and bisexual sexuality definition, while transgender or cisgender or genderqueer as gender definition. But what is a gender identity or gender expression? Gender identity is the concept of a person, of themselves, that can be male, female, a blend of both or neither of them. And when we talk about two-spirit or non-binary person, we're talking about the gender identity. And this is not a common or a recent definition, because two-spirit person is a well-known, acknowledged third gender form for Native American. While gender expression is the external appearance of one gender identity that is expressed through behavior, clothing, haircut, voice, and pronouns. So for the biology part of us, you can think of the gender identity as the genotype of a person and the gender expression as the phenotype. And basically the blend of all these, characteristics is what makes a person. And importantly, especially gender identity and gender expression are not static during the life of a person, not even during the day. Because, for example, if my gender identity or gender expression at work or during the day are more masculine, when I go out in Hillcrest with my friends in the evening or in the weekends, or at least used to, my gender identity and expression moves more toward a genderqueer or less masculine expression of myself. And so this reflects in the pronouns, and I really like this picture that depicts some of the most common pronouns, because it clearly shows that you cannot guess the pronouns to use for a person based on their physical appearance or on their clothing. And especially in such a multicultural environment like the one where we are, you cannot even guess the pronouns to use based on the name, because, for example, Andrea, that is for an Italian person, is a very masculine associated name. In other culture, like for example here in the United States, Andrea is a feminine name, so I would use the wrong pronouns. And how you can normalize the pronouns use in academic setting? It's extremely easy. Just put them in your email signature, add them at the beginning of your presentation in your introduction slide. You can add in the Zoom feature. And whenever you don't know what pronouns to use with a person, because it's the first time that you met them, just ask. The simple way is to ask, what pronouns do you use? So, but specifically in STEM, what are the issues that LGBTQ community face? So there were studies showing that sexual minority students are 7% less likely to remain in the STEM field, and because of this so-called leaky pipeline. So in this survey from 2018, you can see that 40% of the sexual minority students, in this case were only men, left STEM compared to the 26%. And honestly, I do refuse to believe that is because LGBTQ students are less gifted in the STEM field. So obviously there are some obstacles that we are facing. And you can see here, this is a survey from the American Physics Society from 2016, but a similar survey with very similar data came out last year from the United Kingdom Physics and Chemistry Society, in which you see that members of sexual minorities face or at least observe exclusionary behavior. And especially our most targeted siblings of gender non-conforming and transgender, they face even harder life in the lab. And these struggles, these behaviors, are everything coming from sexual harassment, homophobic comments, um, expectation of incompetence, stereotyping, not having the proper toilet facility that someone felt comfortable and safe in using. And even if I didn't have experienced this exclusionary behavior on my own, I have observed in my previous institute in Germany, where a PhD student was kept himself in the closet because his PI was sometimes expressing homophobic view. And and I still shame that I didn't say anything at the time, but I wasn't in the position of doing something. So if you want to have an idea of how to make the workplace more welcoming for LGBTQ scientists, you can read this article that came out last month. That is an interview with 6 LGBT scientists. But the bottom line was, if you see something, if you hear something, say something. Bring the issue immediately up. If you're not feeling comfortable, bring the issue with HR. If you are at the SBP, send an email to the Education and Training, Diversity and Inclusion Committee, and there will be the email at the end of the presentation. And if you are in the position of to be a role model, be a role model for the younger scientists that you wanted to have the role model. And I'm pleased to see that there were a few faculty members and few that are in the position of organizing the conference. And when you organize the conference, have you ever think of the location? Because not everywhere in the world LGBTQ people or other minorities are supported or are protected. For example, in the field of the conservation and ecology, over the past decades, so we are talking from 2020 to 2009, we are not talking from the 50s, 60s, or whatever in the past, over 40% of the conference were held in places that had discriminatory laws against LGBTQ people or other minorities, meaning that you are excluding a huge part of the scientists from going to a conference. And I would honestly I would never go to a conference in a country where I don't feel safe. And doesn't matter how important this conference will be, or for my career, for my network, or maybe the most important conference in my field, I will miss it because I would rather stay at home and be safe than go to a country where I will not be feeling safe. And finally, even if I know that I spoke mostly about LGBTQ issue, I would like to conclude with the concept of intersectionality that was developed in the 70s by some Black professors, and its term was created by Professor Kimberly Crenshaw, and you can read the definition, but I think that this sentence summarized the concept pretty well. If you are standing in the path of multiple form of exclusion, you're likely to get hit by both. And again, while I was not hit by the exclusion because of my sexual orientation, I was being called the lazy Italian, or I was told that as a foreigner PhD student, I was stealing the position to German students, and you know, all this kind of stuff. So if you have to fight, we have to fight together, because if we don't do it, and if we divided ourselves, it will be easier to take away our rights in not only in STEM but in any field. So finally, I just want to share here some online resources focus on LGBTQ issues and some organization of scientists. So you can have a look, and if you want the list, please send me an email. I will share. I will be more than happy to share with you. And I will finally thank you for listening, and thank the member of the Puri Lab, especially Lorenzo, that is a very supporting mentor in for every single aspect of our life. The Education and Training, Diversity and Equity and Inclusion Committee. You can see here our email, that I'm proud of being part of at the Sanford Barnum Prebys, and our collaborators. And if you have any question, please ask.